With the rapid increase of the Earth's human population over the past century, light pollution has become an environmental problem of global proportions. Since humans are poorly adapted to finding their way at night, they adapt their surroundings to them by installing many artificial light sources. The problem is clear when looking at heavily urbanized places like the city of London, the Australian Gold Coast, or other densely populated areas in Western Europe, such as the Netherlands. Many moths are attracted to light, causing them to remain in close proximity of light sources. Since bats are major predators of moths, bats are directly as well as indirectly affected by anthropogenic light sources. Recent studies have shown that artificial light has long-term consequences for multiple taxa. Moths in particular were shown to have lower reproductive success. The animals are distracted by the light and won't feed nor breed near them, reducing the numbers near heavily lighted areas. The effect of artificial light on bats varied. In some studies, the bats avoided the light, while at other times the bats were attracted to the abundance of prey in the vicinity of the light source. This takes us to our research question. What is the influence of red, white and no artificial light and predation risk by bats on moth abundance and behavior? To answer this question, a correlative field experiment and manipulative lab experiment were carried out. We hypothesized that artificial light and predation risk by bats affect abundances and behavior of moths. Artificial light is expected to negatively influence moth behavior. This effect can be quantified with traps we use to catch the moths, a light trap, consisting of a cloth with a light bulb shining on it, and a food trap consisting of a small table with a sugary solution combined with pieces of fruit. It is expected that the moths will be attracted to the light and thus be found more on the traps. The expected relative proportions will be different, as is shown in this graph. The proportion of moths on the food trap is lower in the light treatment as moths are distracted or blinded by the light and feed less. Bats use echolocation to hunt. It is expected that this echolocation influences moth behavior, causing moths to seek shelter. We expect this to result in less moths on the traps. Predation by bats before or during the experiment is expected to have a negative influence on the number of moths found in the experiment. As we cannot measure predation itself, we measured the effect of bats on behavior and predation by measuring bat echolocation calls. The number of bat flybys was measured. If there was more than 3 seconds between calls, it was considered a different flyby. Habitat could also influence our results. For example, differences in vegetation structure can influence the effectiveness of bat predation. Moths may also be more able to seek shelter in certain habitats. Food availability, as an example of a habitat-related factor, can simply influence the number of moths found in an area and thus the number of moths we found in our trap. Vegetation structure is accounted for by using a fixed distance of 5 meters to the forest edge. Food availability was not measured as this was deemed too difficult and too inaccurate. Moths can fly large distances and different species have different food sources. Any habitat effects can be checked using the factor site, as we used a fixed set of locations for the treatments over which we randomized. We also randomized over time. Finally, there are also weather variables, which were measured or later determined. These are cloud cover, moon brightness, temperature, air humidity, precipitation of the day before sampling, wind speed and wind direction. A possible confounding factor could be daytime predation. Time of day could also be important for our measurements. In general, moths are known as nocturnal animals, so time of day is important for moth activity. In our first lab design, the effects of different light treatments and bat echolocation on giving up time of moths were measured in a controlled environment. The giving up time is the time a moth spends on a depletable food source before it will continue to search for another. Other factors that could affect the link between the treatments and the response were food availability, time of day, enclosure setup, lab-reared or wild moths, and the sex of moths. Unfortunately, the wild moths did not react to any stimulus, and the lab-reared moths were not available. A new experiment was needed. And here it is. Bat sound and light treatment both influence the moth behavior in boxes. The sound of bat echolocation is expected to negatively influence moth behavior towards a box, while the light treatment is expected to attract moths towards a box. Additional variables are the species of moth introduced in the experiment and the number of moths introduced in one iteration of the experiment. Some species may react more strongly to bat sound or light, while a larger number of moths may incur some sort of group pressure. Finally, the weather during fieldwork influences these two additional variables. Bad weather means less moths can be introduced in the experiment. In the figure, notice that the colors have switched for with bats and without bats, and that the order of treatments is different on the x-axis. From the figure it can be seen that we expect the least moths in the box with no light and with bat sound. The field experiment was carried out in a wet grassland valley west of Renkum, called the Renkumsbeekdal. This rich grassland is bordered by deciduous forest on both sides. We selected 12 plots along the western and eastern side of the valley, all plots being 80 meters apart from each other to avoid pseudo-replication and 5 meters removed from the forest edge to reduce habitat variation between plots. After performing a pilot study, we noticed that there is a moisture gradient in the study area from west to east, which led us to divide it into two blocks, one in the north and one in the south. This was also a practical solution for sampling because there are only 2 points at which the stream can be crossed. The order and location of each treatment will be completely randomized for the 2 blocks in groups of 3 treatments, making sure all different orders of treatments occur and the treatments are spread over time. Every night before sampling we prepared our equipment at a nearby parking location. We placed our self-made light trap on a camera tripod for stability and prepared ourselves for a cold night. The light treatment that was applied in the field consisted of white light to stimulate most used streetlight color, red light to look at the effect of longer wavelengths, and a controlled treatment using no light. Every night, 6 treatments, randomized across 6 locations, were sampled. Each sample consisted of 30 minutes of exposure to the treatment before moving to the next location. A portable generator was used to supply electricity using a 40-meter cable. In each location, temperature, starting time, wind speed and cloud cover were measured using a measurement device. During the 30 minutes, all passing bats were counted using a bat detector. We noticed that sometimes some gigantic bats flew over. After 30 minutes had passed, all moths present were counted and the setup was moved to the next location. Usually the first moths appeared after 5 to 10 minutes of waiting. Based on a specialist's advice, only large moths were captured for use in a manipulative lab experiment. Our lab setup consisted of a central box connected to 6 other boxes by tubes. These boxes are appointed a random treatment every time. The treatments are white light, red light and no light. This is combined with the presence or absence of bats, which was generated by a laptop playing the bat echolocation recordings. The moths are then left overnight. Every box was provided with 2 types of food, namely sugar water and delicious honey. The day after a successful sampling night, captured moths were released in the central box. following day, we checked which boxes the moths had moved to. The moths are then recaptured in the lab and then released back into their natural environment. Moth and bat abundances are represented as count data. This means that they are Poisson distributed. To perform a statistical analysis, we opted to use a generalized linear model with a Poisson log-linear link and backwards selection method. First, all scale variables were checked for collinearity in R. In this graph, we only show the relations between variables with the highest collinearity values. Temperature had a very high variation inflation factor of around 13. This is not so surprising since moisture and temperature are climatic factors that change in time. After removal, all variance inflation factors were below the threshold of 3. Next, the generalized linear model was performed. But before we can talk about the results, there is one more thing we need to check: overdispersion. The deviance and Pearson chi-square of the count data regarding moths did not show overdispersion, while the bat counts did. This means that a Pearson chi-square scale parameter method had to be used to correct for this overdispersion. Now we come to the part you all have been waiting for, the results. For moth counts, the following variables were significant: treatment, precipitation, clouds, and moisture. For bat counts this was plot, wind speed, and clouds. Now I will go further into detail and show how each significant variable influences either bat or moth abundance. When interpreting these following graphs, it is important to understand that it is the combination of the significant variables that explains the variation. I will start explaining each significant variable for the moth abundances. Treatment is up first. As you can see, the abundance of moth on the light trap is highest when a white lamp is used, followed by the red lamp. So we can conclude that the control is the best condition for moths. The next variable is clouds, and for this variable 5 categories were made, ranging from not clouded to completely clouded. We expect that outliers cause the high difference between cloud conditions. This means that on average there is a higher number of moths when cloud cover is higher. Relative humidity is the next in line, and again we think that the outliers are the cause for significance in this test. An alternative explanation could be that there is an optimum humidity level for moths around 70%. The last significant variable for moth abundance is precipitation. The graph shows a clear trend that moths are higher in abundance when no rain fell prior to the field experiments. Since we applied a treatment in the field, we wanted to show the effect of the treatment on bat abundance as well. And as you can see, the light treatments do not have a significant influence on the bat abundance. The distribution of bats in the area is not homogeneous, as this graph clearly shows. Plots 1, 2, 11 and 12 show in general higher abundances of bats than the other plots. A reason for this could be that these plots are by coincidence close to bat resting places or bat flying routes. Next up is clouds. Again we see a higher abundance of bats under clouded conditions. The last variable in our significant model on bat abundance is wind speed, showing that bat abundance is negatively correlated to wind speed. To end the statistical analysis of the field experiment, a power analysis was performed. This was done by increasing the amount of replications using values from the existing dataset. Currently, 3 to 4 variables can be explained with this dataset, but if we wanted to have shown more significant differences in the count data for, let's say, 7 variables, a total of 270 replications should have been made. We only had 8 measurement days, and each measurement took us at least 30 minutes, and this would have been an impossible task to achieve within our group. The data is again count data, without a normal distribution. In this case, we used the chi-square test with a post-hoc pairwise comparison test to analyze the data. Bat sound is the first factor that was analyzed. The simulation of bat sound did not have any effect, since the same amount of moths were found in boxes without sound and with sound. For the different light treatments, however, a significant difference was found in the amount of moths found in the boxes. White light differs significantly from no light and the central box, while red light has no significant difference with any of the other treatments. Again, we end up with a power analysis to see how many samples it would take to get significant results in a follow-up study, and it seems that around 25 samples are enough, so therefore our setup with 28 samples is sufficient. We are almost at the end of this journey. The only thing that awaits us is the discussion. Not all goals could be achieved within the given time. During this project we encountered several problems. Originally, the goal was to measure the giving up time. However, in our first lab experiment, the wild moths did not show any reaction to the treatments, so no giving up time could be measured. This forced us to redesign the lab experiment. In the field, the food trap did not attract any moths. We believe that our food source was not attractive enough and that the exposure time of 30 minutes is too short. The biggest problem, however, was something we could not control: weather. 4 out of 9 sampling attempts failed due to the weather conditions being too wet, cold or windy. This can also be seen in the result of our analysis, where climatic factors play a significant role in the abundances of moths and bats. Precipitation had a negative impact, while cloud cover had a positive impact on moth abundances. In the literature, however, we found that cloud cover should normally reduce moth abundance. We believe, though, that due to the colder spring temperatures, these clouds acted like insulation, keeping ground temperatures higher compared to clear sky conditions in the same period. This theory also applies to the abundances of bats, where the same positive relationship with cloud cover was found. If this research had been conducted in a period with a more stable and suitable weather, the random variation in the dataset would have been less influential. The bad weather conditions did not only affect the number of captured moths, but also prevented us from working effectively in the field. Despite the weather influencing our project so substantially, a significant result for the light treatments was found in both the field and the lab experiment. White light in general attracts more moths than red light, while red light attracts more moths than the control. Around 50% of the moth species can detect bad echolocation calls and react to them, but studies have shown that artificial light can impede this ability, increasing the capture chance by bats. Other negative effects range from reduced reproductive success to changes in foraging behavior. This brings us to the societal aspect of our research. We advise the following possible measures to reduce light pollution. Our experiment proves that red light, although keeping the environment lighted, is the least disturbing for moths. Other alternatives are the use of streetlights that concentrate their light downwards, using less lights, or wherever possible to remove lights. We thank you for viewing and hope you enjoyed it.